No.
No?
No. I don't think so. That's a rare fish that doesn't just run away.
Yeah. It's got too much stuff hanging off him.
Mm-hmm. There was one up there, but it got, oh, there, a larval macro, or maybe it's attracted to the lights. It's gonna come in the jar.
Had enough of this guy.
Mm-hmm. Sure, yeah. I mean, wow, it's pretty cute. I just really wonder how these things came to be.
Yeah. Strange. Close up of this guy. It's going to be fast. Not too fast. fish. Oh, that's a better one.
Let's see it, yeah. Maybe we take that one.
Shots first, glamour shots.
Mm-hmm. We maybe have been getting some of those in the.
Yeah, it's better.
Wow.
You want to try and capture this guy?
Sure, yeah.
Yeah. Challenging one.
I think it's challenging.
Yeah. Like a challenge though.
I don't know what kind it is. But Delano said he wants jellies.
We'll want to try and get this one in the blue one maybe. I don't know. Well, we can try it. Just because he's so big, he might come out.
Helmet jelly, maybe, huh? Hmm. Oh.
This one actually is forcing the water away, so maybe we'll go for one. I think that blue thing's pushing the water.
There he is again.
Yeah, a little bit. He's swimming against it as well.
It seems like there's some current now.
Coming from the bottom.
Oh, it's right underneath it, yeah. It's about to go in. Oh, nope. It's almost in. It's right underneath it now. Yeah, it's in there. It's in there. Oh yeah, it's in there. But it's not. Oh, it's closing slowly. It's still in there. Oh my God. Did the bottom close? Did the bottom close?
Yeah.
Oh my God.
That was like nerve-wracking. Oh well, we got him.
Jellyfish.
Well done.
I was holding the thing.
It's
That's
not
the number
closing.
one.
Oh wow, that's good. Yeah, I think
You see the
with that,
latch?
the blue thing.
Ah,
Oh yeah, the latch. Is it closed on that thing?
no, it's not.
I hope it is in the bottom.
Oh my
Yeah,
god.
yeah, I think I'll survive.
Yeah, you think like that.
The filter thing, when I was trying to come up, it was like forcing a bit of the water.
Maybe the
Up.
holes need to be bigger.
I mean, with good timing, we don't even need that thing on. Right. Okay. Nothing hunter mode.
That woke me up a little bit.
All that excitement.
Yes.
It's not always it's not bald.
What's that thing? Ouch. What time is it now? It's 4.
Jelly there. Same one from. Same type. Get in the shot set.
I don't know.
Same one. One shot's better. Yeah.
Oh wow, look at that. Same kind, yeah. Let's not take that one.
Yeah, some. Oh no!
That looks like a big
Finger
one.
slip.
Is that one even bigger?
Now we need to do it. Now we need to do a white balance. Ooh. My finger slipped. There we go. Yeah, my finger slipped. I pressed the wrong button by accident.
So that's your little way of fixing the cameras.
Yeah. There's something there. See it?
Mm-hmm.
Yeah, this looks cool.
That's one of those butterfly things, isn't it? Or no?
We bump up another.
Wow.
ISO. Notice that on the stream. carry on.
Mm-hmm. These are cool. We've seen a few. They're kind of amazing. Cute neck. Like a couch in here, like a little cot for napping.
I think you get folk sleeping quite often if I had one of them in here.
I know.
Oh, look at this, are you not interested?
Oh, we don't want to collect it, but if you want to look at it, you could for a minute. That's a pretty long one. I think that's another siphonophore, right?
Yeah.
I don't know. I'd like it if we could see some like foraminifera or something, but I haven't seen too many of those. Wow.
Picture this one.
It's just hanging out. calibration.
Yeah.
My goodness. I mean, wow. That guy's not really expecting anybody to come nearby. He's just thinking he can hang out with all that stuff.
It's just chilling. It's massive actually if you're
What in
looking
the
at it
world?
camera.
What?
You seeing that in our sit cam.
Is that a ctenophore or what is it?
It's a ctenophore, yeah. I figured the best lighting for you.
It's just crazy. It's almost like it has roots. You know? Pseudipid ctenophore. Yeah, look at that. What does it do with those long tentacles? I mean, that's wild.
I don't want to get too close to them because I don't want to get those tentacles trapped up.
Wow.
Try to get a bit
I
closer.
wonder if they can grow them back.
It's been maxing anyway. As far as I live enough.
Oh, I'm getting some. What? That was a baby. Oh, that fish was a baby rat tail or granadier. A baby of the genus Lazumia or Coryphaenoids. Huh. Getting some. Ben. Oh, okay. Thanks, Ben. We've got our fish guy giving us a little bit of a
This guy. He's over there.
wild. There's another ctenophore. And there's a fish. Yeah, we're getting up into some.
So like organisms like that, like when you're catching a net, you wouldn't really pursue because it's too soft, right? Like you just
Have you looked in this lab at the samples we're
No.
bringing up? You should go in there.
so we do have those things.
Which ones?
Like the ones that what we just saw.
I mean, we
It
get a
seems
lot.
too soft.
We get a lot of ctenophores, and yeah, I think their tentacles would maybe break off, and they don't look so like luminescent in the lab, but they
They're just
do.
more
They do
gelatin.
sparkle a little bit,
Okay.
especially if you shine a light on them. So, yeah, we get a lot of ctenophores. but they don't look nearly like, I mean, they just don't look as cool,
Yeah,
right? It's
yeah.
just, they just look kind of like a blob of jelly. You're like, oh, okay, whatever, it's another ctenophore. And
Yeah.
then you see them in their natural environment, you're like, wow, that's incredible.
You're like, wow, that's a ctenophore.
Yeah. I mean, some of them are more beautiful than others, you know. We get them near Seattle in the Puget Sound. They don't have all that, you know, decoration, and they're not, I don't know, they don't seem as interesting as these deep sea ones. Yeah, I got a message about that fish with the long tail, that it was, some of them people heard me say it's a baby rat tail or grenadier, baby of the genus Nezumia specifically, or Coryphaneoides. I can't even say these words. In case anybody's interested. We have, as I've said, people on this ship that really know their taxonomy, and they're all working really hard in the lab to sort through all the animals from our net trawl. I think they're getting close to processing this net, but we might put the mock nest in again tomorrow night if we're still here, if the weather hasn't chased us away.
Yeah, because Friday's supposed to come up.
Yeah, but you should definitely go check it out. It's pretty cool.
Yeah.
Tony came in, looked around.
But they're all done now.
No, they're still in there.
They're still in there?
I mean, I think they're still in there. I mean, they're getting close to getting done, but there's 6 nets, so they were, you know, they go through them one by one and sort every single animal, and
This guy.
they weigh them and they measure them and they identify them under the microscope.
Do my ISO.
Look at that thing. I mean, they sit there. It might take them 5 minutes to identify one, you know, shrimp at the species level,
Oh, well, it's fast.
or a fish at the species level. They could sit there and count, you know, all the different parts, measure parts, all that to get the identification.
So it's very interesting, this cruise, because it seems like you guys are processing it more than previous cruises. A lot of times, like on previous cruises, it's just like, we'll process them when we get back to shore.
Right.
And you guys are actually doing it kind of immediately.
Yeah. Well, I mean, my lab is going to be doing these, you know, measurements of the metabolites in them. So that's like basically going in and taking samples of them and saying, like, when you go to the doctor and they measure your cholesterol or your blood sugar or your those, they will measure all these things in their gut mostly, but also in the animals, and try to find out what kind of, you know, metabolisms are happening in the animal gut. And so those samples have to be processed really fast.
Yeah.
So we either have to freeze them immediately or, you know, dissect them and freeze them immediately. And if we freeze them, they're harder to dissect
Yeah.
once they've been frozen. So we really are trying to get fresh specimens. And I think the same thing goes for the net. It's like when they're counting all those organisms, you know, the jellies don't preserve well,
No,
right?
no,
So
yeah.
like all these things, they need to kind of get certain things when they're fresh in order to actually count and measure them. But it's quite a production in there. Like,
Yeah.
it's incredible seeing this bin of like hundreds of things come out in the little trays, counted, weighed, measured, named. You should give it a number.
Wow.
Yeah.
The top latches are closed on that one.
It's kind of amazing.
Especially since we did nets back to back, I guess. Yesterday.
Yeah, so they've been awake for more than 24 hours. I think a lot of them with just a little bit of a nap in between.
Hand over time.
Oh.
originally to see how it worked, but I think it was forcing too much water up. It wasn't like when I was coming up underneath it, it like blew it over the side. So if anything, bigger holes are just keep it out because obviously don't need it. I don't think we need it anyway.
Got Leonardo back.
Right, adios.
Yes.
For an hour.
We're just kind of slowly making our way up and stopping for things. And we collected a few jellyfish in the jars. Look at that one. He's pretty big. I guess we've gotten some of that kind in the nets. Actually, now that I'm looking at it. Look at that thing. Oh no, it's another one of those. Wow. I didn't think that's what that was. You can keep going. We're planning to stay down until 8 o'clock, so we have 4 hours to go. I mean, we'll come to the surface and be on deck by 8 o'clock, I think. Or no, we'll be pulling it out of the water at 8 o'clock. Or at least that's the plan. Did you guys ever get a dive plan from us? I guess maybe Colleen sent it to you. Good morning. Wow, you're up early.
I got to sleep.
I wasn't quite expecting you this early.
How's it going?
What?
How's it going?
Pretty good. Yeah. I don't know. We put some things in jars for Delano.
Nice.
I
What'd you
don't
do?
know anything, but well, we got this one that's
See ya, yeah.
in there.
Are they all jellies?
No, I got one
Theodarian? Oh, cool.
Uh-huh.
Like a really big one?
I don't know. Yeah, it was a while ago, so we'll see how it lasts. It was down at like 1,500 meters.
Cool.
Yeah, we got a Theodarian. We got a this, I don't know what kind of that last one was. We got a helmet jelly, I think, in one of them. We got yeah, some other things like that. We saw a larval ratfish, or a baby
Rat tail?
rat tail
That's cool.
that was really like just hanging out for a long time. It's been all right, you know. We're just very slowly. We've been just very slowly, steadily coming up as we explore.
Cool.
Oh yeah, there's a solmesis in there, in one of the jars. And we did see some pretty awesome ctenophores. Oh, we saw this thing. It was completely clear.
Oh, an amphipod.
That's what I thought. It was maybe an amphipod.
Yeah. I don't know the species, but I think it's a parrot, but no, it's not. It's definitely an amphipod though.
Yeah.
That's really cool. Was it big?
It was not that. It was that clear amphipod. It was. I don't know how big it was.
Okay. That's pretty big for an amphipod.
This is it.
Whoa.
Squid.
Yeah.
Yeah.
Nice.
Oh, there's the rat tail. We didn't get a very good picture of the rat tail, but I got a better one. The rat tail.
Oh, that's so cool. Oh my
Yeah.
gosh.
It was pretty big. It was, even though it's a baby, it was
My
pretty big.
grad school advisor's rat tails are his like favorite thing when he studies, so he would be very into that.
I mean, this ctenophore is kind of spectacular.
Weird something's happening in front of it. Nice.
Um,
Oh, there's a lot of stuff for 800 meters.
I saw that pretty early on. I don't think we collected that. That was before I knew Delano really wanted that type of thing.
How many jars are empty?
2. I mean, we could empty. I don't know. We could do something with the other ones.
No, I think if he's looking for stuff, that's definitely a good
I mean,
priority.
And that one on number 1.
Oh, wow.
That's something
big.
we've gotten those in the nets already, and I don't know if he was given any or what.
This would be probably in better condition. And it's always nice to get the like image, I think, to match to it for ID.
Mm-hmm. I don't know. I assume it's got some kind of gut.
I mean,
Mm.
kind of.
Yeah.
It's like kind of just a simple like cavity for them. And they don't seem to have stuff inside of there frequently, but you can still like get that part of it and look at the microbiome if you know what you're doing, which he does.
Yeah. All right. Well, I might go to sleep since you're here. Are you ready
Yeah.
to take over for real?
Yes.
Okay. I was really struggling there for a while, but got my second win. All the excitement collecting the jellyfish.
I guess just like for how fast to go up. I mean, we're still planning to be on deck around 8. Okay.
Yeah. So
We have 800 meters in 4 hours. Okay.
so every hour you've got to go 200 meters
Mm-hmm.
or more because you want to be on deck at 8.
Yeah.
Now what is that?
That's a siphonophore.
It's really,
And then down left looks like a larvation.
yeah, there are a lot of larvations
Yeah,
down
yeah.
there. Some of them looking like the parachute kind
Mhm.
of, and different shapes. Do we know if there's a Schmidt discord? I don't know about Schmidt discord. Someone's asking if there's a Schmidt discord.
We've been abandoned. We don't have a person over there.
He just stepped out.
Oh,
That was
okay.
Josh. I'll probably be back in a minute. I don't know. Just a little shrimp. I'm getting pretty good at spotting things. My third ROV dive of my life, and I'm getting, to know the deep sea a little better. Boy.
Yeah, I can take a look.
All right, yeah, I logged the things, and I took very messy notes here about.
Did you write on the like printout what
No.
where stuff is?
Do you want me to do it?
I mean,
I should. Okay.
as long as someone knows which jars have stuff.
I
in
just
them.
don't know. Well, they're the ones that are closed.
Okay.
I mean, it's 1,
3, 4, 5.
Okay.
1,
Good use.
3, 4, and 5, and 1 has. I don't know what that thing is called. It's like. It's a. I don't know. And the 5 has the solmesis in it. So 4. 3 has the Phaeodarian area. Number 4 has some sort of a jelly in it, but I don't know what it is. Maybe it's the helmet jelly, maybe. And number 1 is this thing, this
Okay.
crazy jelly.
You can just say Medusa.
Medusa.
Yeah.
There you are, and they're
Thank you.
logged in here.
Okay.
And I tried to write their depths, like number 5. Do you care about that? This
Well, no, just for like when we take stuff out, we
919.
want to know like what's in where.
Phaeodarian was at 950 meters.
Wow.
The helmet jelly. The solmesis was 919. Oh yeah.
Oh, that's a doliolid. It's got all the tail is a bunch of clones
We
that are
haven't-
gonna pop off and become their own individuals.
I can already tell you're gonna do better than I did.
I mean, you've got a lot of stuff to do great.
I mean, but just knowing what things are. Of course, because I explained I'm a chemist. The reason I'm not in the room sorting animals is because I'm a chemist, and I'm
Yeah, it looks like they're
useless
still
in
in
there.
there.
Yeah, I know they're still sorting animals. Thank you. You probably could, if you see something amazing and you don't have any jars left, you could probably let something go. Colleen had said something about collecting that Phaeodarian,
Mhm.
so I did
collecting one, so I did. But
Yeah,
we
no.
only saw one.
Really?
I mean, I'm sure there were others. We just, I don't know. That was the only one we noticed, and we zoomed in on it.
Okay.
And it was way deep. All right.
Good night. Oh, let's get a shot of this larvation that's in the this one. Yeah. Because it's got a live one inside of it. Oh, never mind. We just kind of destroyed it. They're really delicate. It's okay. The animal is okay. Their houses that are around them are really delicate, but they will discard those on their own as much as like every few hours and build a new one. So that wasn't actually like that bad to do. I can introduce myself. We had a little bit of a shift change here. My name is Lee Miller. I'm a postdoc researcher at the Monterey Bay Aquarium Research Institute. I'm also not really too much of a midwater animal expert, so I'll do my best. My focus right now is on the particles that you see all over the screen all the time, and specifically the microbial communities that are in them, as well as what animals produce them. So trying to kind of connect the animals and the microbes through the particles that are produced.
To get the bioluminescent shots right
Yes.
there. I had them.
Oh, whoa.
I just kind of, I saw the comparison. I was like, ooh.
That's so cool.
Because it's really cool. And that's from the sediment traps?
Yeah, so the little picture within picture is showing some of these particles that we collect in these gels. So we have this sediment trap that is out for 3 days. We're actually going to recover them, like our second set of them right after this dive. And we collect a bunch of particles in them, and I was really curious to see if we could detect bioluminescence on the particles. So that's either bacteria or potentially some other things that would be glowing, and that is really important because it affects how they are, like how frequently they are consumed. And so I've been taking pictures of our particles in the dark and actually was able to get some bioluminescence, which is really exciting because there's not a lot of data on sort of the rate of bioluminescence in particles. So that's what you're seeing there. Thanks for putting it together. This looks like a ctenophore, a comb jelly here. Very small one. I'm also seeing a lot of Chaetognaths, which are known as arrow worms. In our trawl nets, we've gotten a ton of those. And they're common throughout a lot of the oceans, but I'm not used to seeing this quantity that we've been seeing on this cruise. So those are these little things that are darting around. That kind of look like fish from a distance, but they're actually worms. It's a siphonophore. And then we've got a bunch of these like low bait ctenophores in view, though the thing that's kind of in the center of the screen now. They're super, super delicate, so we've had trouble collecting them and keeping them intact, but they're really beautiful. That's a little fish. Doesn't want to be looked at.
Weird. Oh yeah, what's this thing? Oh, okay, yeah, this is also a ctenophore comb jelly, really cool one.
What up are you gonna collect it? Niskis?
Oh shoot, I should have asked. Let me see what Anitra wants from that. If she's not already asleep, that's a good point. I'm gonna assume, unless she tells me otherwise, that we'll probably collect niskins at like, well, so we're kind of, the migration, it looks like, is already kind of starting. Well, no. Is the EK80 on? And like showing us data? I know it won't be like great data, but
No, I don't think so, but I can turn it on.
that'd be great, yeah, because we basically on the way up we'll probably collect where the animals are densest, which is probably gonna be around like 500 to 200, but we'll look as we go up. Oh, never mind, she does not want niskins. Okay. So we won't.
Hey.
It's still will be nice to see where the animals are gonna be though.
Yeah.
What's this thing down here? Oh, it's a siphonophore. We can keep going.
Right.
Yeah, it's a doliolid actually, but yeah. So we're currently heading up, but very slowly to look at stuff on the way. So on the way down, we did these video transects, which are very quantitative. So we'll be able to actually quantify the amount of animals that we saw at each depth, which is really useful because the net tows we do are also quantitative, but they're going to be, each one is going to be biased towards different animals. So the nets are where we get a lot of fish that is kind of scared away by the ROV. But then a lot of things that are gelatinous are kind of torn up and aren't in as good condition. Whereas with the ROV, we get really great footage of gelatinous and really delicate things. So by combining both of them, we get kind of a better idea of the whole picture of what's down here. But the thing with those quantitative transects is we can't like stop and zoom in on anything. So it's really nice to be able to do that on the way up so that, you know, we can see a small image of it in the quantitative transect, but if we have the footage from like this, we can be like, okay, that's probably what the species was. So like right here, this is a big siphonophore. And the vehicle is going very slow. Currently, it's like 0.1 meters per second, so we're going super slow just to try to be able to see things. During the transects, we go like just a little bit faster, but not much. We're not covering a ton of ground here. As we get shallower, there's probably going to be more current, which will make it look like we're going faster. Wow. That is also a comb jelly. It's really easy to identify that that's what it is because they make those rainbow sort of like flashes of light. Ooh. Is it?
Yeah. Deep down was way calmer.
Mm-hmm. I'm surprised there's current even this deep though.
It's not a strong current for what we are doing, trying to chase.
Mm-hmm.
It's not. It's too much.
Whoa. That's a pretty large ctenophore. Oh.
This is exactly how they look when they're on deck, yeah.
Yeah, definitely. No, it's actually amazing when you get these animals out of the ROV or out of the net. They're like, all of this detail is completely lost. They're so transparent. It's like they look like just like a blob of goo with no detail.
Yeah, here we can see everything.
Mm-hmm. So it looks like it's maybe like 6 inches across, would be my guess. Very nice. Yeah, we can keep going. So someone's asking about what sort of degrees you need to do this kind of work. I have a PhD, but not everyone on the ship or in this room does, for sure. It kind of depends. So I'm a researcher, a scientist, and I specialize in this kind of deep water work. But do you guys at the ROV want to talk about kind of like what degrees you needed or how you got to what you're doing?
Yeah, I'm a electronic technician, and I've done the start with the ROVs just after it, so I have no other graduation above that.
Nice.
And because the ROV is kind of a niche, so specific that you kind of learn by doing and being on the industry, and then you do courses and stuff like that more directly to the ROVs. So that's how I got into it, and I've been doing since then.
Did you start
years.
in oil and gas?
Oil and gas, yeah. But I've been in projects for wind farms and shipwrecks. Different stuff besides oil and gas, but mostly was oil and gas.
Nice.
That's one of the biggest markets for us.
Oh, too far from the ship?
Yeah. I might ask them to start moving east. This way we can go with the current. It's better
Bridge,
than
are we?
against.
Yeah, go ahead. Hey Tony, can we get a little bit of a movement to the east? Start drifting with the current. Yeah, no worries. Thank you.
Thank you.
So that's a old larvation house that's been discarded. Kind of looks like a deflated plastic bag, but that's the mucus house that they make. And you can see it's collected a whole bunch of particles. And so this is another thing I'm really interested in studying, is the effect that these larvations have on what we call carbon export. So that's carbon dioxide that is fixed and turned into biological matter and then sinks down into the deep ocean. This is one of the major ways that carbon is removed from our atmosphere. So it's really important process, and things like larvation sort of help speed up that process and make it more efficient by collecting all these particles on this one large mucus net, which then when it's discarded sinks much faster than the particles would just on their own. And so it reaches the deep ocean and gets removed a lot faster than it would, or a lot more of it is removed than it would if it was just the individual little particles sinking.
Okay.
Yeah, that's a good question. Someone asked if there's something that eats the discarded houses. There are definitely things like copepods, which will sort of latch onto it and eat it. But because they sink so quickly, relatively speaking, the whole thing isn't going to be able to be eaten before it reaches like the seafloor probably. So it's mainly going to be really small things like copepods and also protozoans, protistans, so like single-celled animal-like creatures that are eating it. And then the particles themselves definitely get eaten. That's one of the reasons that I'm looking at their bioluminescence, because particles that glow are much more likely to be eaten by anything that wants to eat particles, because they're just going to be able to see it better. So I'm trying to quantify like how many of the particles are bioluminescent at different depths and at different locations. And these are some close-up shots of what some of these particles look like. There's a lot of diversity in them. Some of them are like really dense fecal pellets. Other ones are kind of looser aggregates, and all of that's going to affect how fast they sink and how much they're going to be eaten by other things. Yeah, and that picture, that plate shows just how diverse the different types of particles are. That plate was collected over 3 days of sitting out in the ocean. This is another one of those dinner plate jellies. They seem to be really common. They're really beautiful. Mm-hmm.
So these 7 traps that are going to come on later today, are you going to try to do the same kind of bioluminescent pictures?
Yeah, that's the plan. Yeah, so we have 2 different trap arrays out. One of them we put formalin, so formaldehyde, just a little really like low concentration on top of the traps, and that stops any growth, so we're able to quantify like what are the microbes that were on there when the particles landed. But that means that they're not going to be bioluminescent anymore. So the other array is the one that I've been taking pictures of. So it's really great to have like both of these trap arrays out, because the ones with the bioluminescence, they actually keep growing. So I was able to take pictures over like 5 days and see which particles gotten more versus less bioluminescent. So yeah, we let them go from the ship. They have a flag on top and a flashing light, and then like a beacon that allows us to track it. But otherwise, these traps are just like off on their own for a few days, and then we have to go chase them down and recover them.
Yeah, I think right now it's about 25 nautical miles away from us.
Wow, and we've been like trying to stay close to them. They really move a lot. So jellyfish, you're asking if they think, they don't have a brain, so they definitely don't think the way we do. They have what's called like a net neural system. So they like experience sensations and they react and respond to things that are happening around them. It's just not centralized like the way it is for us. Oh, looks like there's a really big siphonophore below us.
And it's spiral.
Mm-hmm. Nice. And these guys, these siphonophores, this isn't actually one individual, it's a colony. And so I think that's that's an even more interesting way to think about like do they think, because it's multiple individuals that act together as one and respond to things together. So at the very bottom there, those are ones that have become swimmers. So they're specialized to be the like swimming individuals to move the whole thing, whereas up higher are the ones that are specialized to capture and eat the food. Nice footage. That's really beautiful.
All right, that's my time.
Okay, thank you.
Okay, you're welcome.
So we might want to start heading up just like a little bit faster. So we only have 3 more hours.
Okay.
It's your shift all the way to 8.
Wow. That's that. I imagine that we won't be coming on deck bang on 8 because we'll need 4 guys that will be until probably around about 8. So we'll either be before or after. Lee, do you know an updated plan in regards to what we're going to try to do today?
So yeah, my understanding is up around 8 for the ROV, and then we're gonna chase down the sediment traps and recover both of them. And then I don't know what's after that.
Run away from the weather.
Yes, definitely. Looks like there'll be one more mock mess to like tomorrow night.
Minutes.
It could be argued if the weather's following this up, we want to get ahead of it.
Let's take a look at this thing. Yeah, do you know if the storm's gonna be, what direction it'll be heading?
It's following us, I
Oh,
believe.
great.
Up north.
Ooh. Oh, nope. Running away. I'm messing with you. Doesn't have a brain, but it's smart. Mhm. A lot of these animals in the deep sea, they'll have like a startle response and they'll swim away for a certain amount of time, but they don't do it for very long because at this depth, normally there wouldn't be really much light at all. So you don't actually have to go very far to get away from like the sight range of a predator. Obviously an ROV is a little different from a typical animal down here. But they also don't have a lot of energy because there's not that much food, so they won't swim for too long. Wow. Oh. Got its energy back. Nicely done.
Right. Let's go.
Ooh, shiny. Are you still zoomed in or something?
Yeah. Adjust the focus a little bit.
Hmm.
That.
Yes.
not in autofocus just now because it's easier to see stuff.
Mm.
Is that good better?
Yeah, I think that's good. So it looks like from the EK80, the migration is fully done, so they're probably going to be the most animals around 500 meters.
Okay.
So,
500.
yes.
Low speed.
No. The slow speed up to 500 is good. But then once we're there, we'll probably want to hang out there for a while because I'm also imagining above 500, the currents will be
Yeah, we don't want
too
to be
intense.
too shallow.
I can't see the scale now, but is it around 500 that it starts getting strong? So,
Starts picking.
Yeah, I would say 500 just starts to get.
so do you want to go deeper than that, or do
No,
you
I
think
mean,
500 will be okay?
It actually looks probably more like 400. I think if
Okay.
we stopped at 500, it would be good enough.
Okay, that sounds good. Oh, there's a fish.
Looking too.
It still feels like the zoom is maybe a little close in the main camera.
It's all around.
Oh. Or not the zoom, sorry, the focus.
That's great.
Oh, that's a larvation without its house. Naked one. What was that thing?
Oh, that's good.
Oh. Mm-hmm. Yep. So as this is another discarded larvation house. As they sink deeper, they get this sort of long, elongated shape. Or actually, no, that might be a radiolarian. Yeah, I think that is another radiolarian. So that's actually one really large single-celled organism. Wow.
Yeah. Speck of dust.
It's got detritus stuck to the outside of it that it's probably feeding on.
Do you want to catch it or you don't want to catch it?
No.
Oh.
Here's a siphonophore in the background.
No, I don't want
This one you mean?
oh, there's two.
Yeah. The lower one's the siphonophore. I don't know what that thing in the back is. It might be another radiolarian.
What do you want to look at?
The thing in the background.
It's leveled up.
Yeah.
Let's go down first. I think it's on the other side. I think it's just a bit higher.
Whoa. Okay, this is siphonophore. It's just got really long tentacles. Ooh. Very cool. Ooh.
Open to this. Okay. See ya.
Oh, that's a delicate one. Oops.
See you.
The good news is these guys are colonial, so
Yeah.
they can just form new colonies.
Some of them don't like the light at all. Some of
Yeah.
them, as soon as they hit the light, they're just.
Interesting.
I've seen that a few times.
Huh. There's
yeah,
another
whatever
piece
you
of.
gotta tell yourself.
It's definitely not the
Just
props.
Yeah, it's the light. The light definitely. I think it's.
like that was definitely Tristan that time.
That time was just rusty for sure.
Whoa. We got a lot of shrimp in the nets. It's really cool to be able to see them up close. because they're really beautiful, but they're really hard to see with the ROV. They're fast.
Mm-hmm.
There was one that had these like bright blue antennae, or like fluorescent.
Yeah, that's good. That's fine.
Oh, let's look at that.
Creature. Yeah.
I think it's another teen. Nope, that's a Medusa. Another one of those dinner. Well, might be the same kind that we chased. Yep. Yep. Oh, that one's a Tinaphore. Pretty. This one is extra sparkly.
Oh, squid.
Oh. It's funny, they like kind of come from behind.
Sneak attack.
Yeah. Probably works on fish.
Not so good with the
No. Like, I feel like I see them all the time in the like rear facing camera, but almost never in front of us. Oh.
There he is.
Yep.
It is.
Oh, hello.
Oh, sorry.
Excuse us.
Shall not.
I'm
Jelly
not
jar?
This guy might be a little fast for that.
Challenge.
Oh,
Bye.
Yep.
It's so, yeah, it's so gunky. Like, you think it's like a liquid, but it's really not.
No, it's like skunk.
Mm-hmm.
He's skunk of the sea.
now?
Ooh, big.
Just the non-CCTV.
What's he asking about?
Good job.
It's not responding.
No, it's wrong. Like the address.
Reboot.
Right there.
Power out, power in.
The ocean is kind of full of snot, mucus. It's like a lot of these things produce a lot of mucus and jelly. Oh. What's that?
Nothing to concern yourself with. No, it's just the alarm on the HV.
The what?
The hydraulic unit for
Oh,
the
okay.
large handling system.
Ah.
Someone gave it a little bit too vigorous. Command.
Maybe it's because the battery was holding the like stick.
Uh-huh.
What's this red thing? Probably a shrimp.
No, I should. I'm sorry?
Oh, sorry. I was just wondering what the red thing is, but it's a shrimp.
Oh. Yes.
So we have a number of Spanish speakers and people from Latin America on this cruise, but they're all really busy right now, sorting animals that come up in the nets and identifying them. So unfortunately, there's no one in the room right now. Most of our team is currently in the other lab, identifying, weighing, and photographing animals.
That's what I was saying.
Are we registering you want to stop at that 3.5 or keep going?
No, we can keep going, Tony. I'm fine. Cheers.
Okay.
Shrimp. Pom-poms. Yeah, let's zoom on one. I think it's those same forams. I just think they're really cool. Very nice. Photobombed by an amphipod.
Photobombed by the amphipod.
Come on, guys. Trying to look at the single-celled organism.
Some people.
Okay.
Yeah.
Oh yeah.
Bet Jelly's still alive.
Oh yeah.
He likes it there.
Yeah.
Definitely. Oh, fish.
Fish. That's fish. There's fish there, dude.
Did everyone see the fish?
What? Where?
Just it?
Nah.
I was gonna try to convince us that was a switch.
Ooh, Xyphenophore.
It's me, Andy.
I hate when you're hot.
So we can kind of just like hang out around
Yeah.
here and just
Yeah.
keep flying around. It would take you guys like about a half an hour to get to the surface from here.
Yeah, it takes like 20 minutes to do the float arrangement and stuff.
Okay.
And we're gonna need a full team down here, so obviously the morning meetings going and breakfast going.
Mm-hmm. So
probably
after, it would have to be after 8.
I think so, yeah. If you
Okay,
want to stay
that's
till
fine.
8, it'll be shortly after that, but I mean we can almost get there.
That's what's on the plan, anyways.
Yeah, no, that's fine. Just trying to get an idea of the timing.
Wrong way. I think we're going for it. No, we're getting closer now.
Well, then it must clear, right?
Up that way, please.
Swimmer.
Fish. It's like I wish the fish would let us zoom on them. I think that's a cyclothony. Yeah. So that's like the most abundant vertebrate on the planet are these little fish called cyclothony. Mm-hmm. Nice. They're also called bristle mouths. They've got like really sticky teeth. We get like hundreds of them in the nets.
Ooh, big fish.
Oh, nice. Where? Oh, below us, I see. I don't know what kind of fish that is. but it looks like a migrator. Because, yeah, the ones that are fast like that means that they're migrating up to the surface at night. The ones that are residents down at these depths tend to be a lot slower. Ooh, hello. I would be very impressed if we could catch that in a jelly jar. Is the arm still not working? At the very beginning of the dive, the arm was having issues. They were.
Well, that's on a rail, so we can put it out.
Oh, you don't use the arm for the suction?
Don't need to.
Don't need to, no.
Oh.
We can try and set up another one.
No, that's okay.
Just like that. Put it out there.
We get plenty of fish in the nets. Oh, nice. Yeah. It's gonna swim right in.
Sorry.
That's cool. I actually don't know what kind of fish that is. So this is where all those, or a lot of those migratory fish are hanging out during the daytime. That's kind of why we're hanging out here, because this is where the largest density of animals is. So many amphipods. Seems like they're attracted to the light. Just getting photo bonked by everyone. Shrimp. Be cool to get a zoom on them if they don't go way too fast.
Sneak up on 'em.
With this big, bright, loud thing. Yeah. Nice.
Stopped.
They're too smart for us.
I'll swim out a little bit.
Mm-hmm. You said you wanted a challenge.
Yeah. I'll just put them over here in this tube. Alright, feel good.
Mm-hmm. Some people in the chat have been mentioning these foraminifera, which are the kind of fluffy pom-pom things. Those are also single-celled organisms, where they've got those kind of spikes coming off of them. They're really shiny. Forams are really cool. There's benthic ones on the seafloor, as well as these pelagic kind that are in the water column. And they've existed for a long time, and so they're actually used by a lot of scientists. They're like fossilized bodies to record past ocean conditions. So they're made out of calcium carbonate, and there's a lot of different things that you can learn from the makeup of the calcium carbonate of them. Ooh, there's another fish. So you can learn about like the temperature and the amount of carbon dioxide that was in the atmosphere millions of years ago just by looking at really old forams. So it's cool to see living ones like this. So are you mostly drifting with the current here?
It's actually going a little faster than us right now.
Okay.
So, far ahead.
Nice. So that in the center of the screen, that's one of those forums. No, it's okay. I was just for the audience sake, because I was just talking about them. like whenever we stay in one place for too long, these copepods just like swarm us, or not copepods, amphipods. What? It's this little thing. Oh, way too small. Yep. Off to the right is something bigger.
To the right.
Talking about this thing. That's now running away from us. Never mind. I don't know where it went. Yeah. Okay.
All right, mate.
Okay.
It's just like weird to start, but I noticed the patches now done on the top of that one. It was only one in. So it must have sell off, yeah. But we've got, I don't think this is a good thing. No way. Because I tried to get jelly that's in number 1 and number 2 first, just to try the theory that it like pushes the draft that it like displaces the water and they're easier just to keep it like that. I think it would then use everything that's sold. You want to then fly up like that? What? Leo ain't flying. Oh, Leo ain't flying, I'm flying. Oh, you're flying.
I think you said Leo. You're just going right on out.
a really large larvation house.
Do you want to see it or you don't want to see it?
I'm just.
I think we're just gonna shred it, although maybe not. Yeah, there we go.
Yeah, once it gets caught,
Yeah.
thruster wash. As soon as
Mm-hmm.
you're thrust away from it, it's not gonna.
Yeah. I think the jelly jars would have to be much farther out to collect them.
Yeah, you maybe get one in there, but would it stay intact? So,
Yeah, the jars would have to like
one little
really,
touch.
really be sealed.
Yeah, one little touch off the side of it, I think it would
Mhm.
disintegrate as well.
Yeah.
This should be the easiest depth
Mhm.
just now, I think.
According to the EK80, yeah.
Yeah.
We don't have to stay like exactly here. We can, go up or down just like a little bit.
Yeah.
We just don't want to get back into that strong current if we can avoid it.
Mm-hmm. Well, we're going to hang out here for a little while, but then if we come in shallower than this, we're going to have to turn on out haulers and stuff, and then it's getting up a good bit as well. So
The species, they probably are deep enough. It's clear they then likely go to the depth.
Yeah, and I think this is the depth that they're hanging out at because you can see on there that big density is around 500, a little bit above.
Yeah, they travel down and stay around there. Because above that, they're not. They can be seen.
is it like
Mhm.
temperature?
It's the
or
light.
The amount of light's too much.
Yeah, I mean, they can handle that light. It's just that their predators can as well.
Yeah.
So they hide down here. Ooh, there's something. Oh,
Down here?
Yeah, it was right there. We go. That really transparent thing. That's a
Okay.
leptocephalus eel larva. We saw a bunch of them in the surface waters at the beginning of the dive, so it makes sense that now they'd be down here.
I'll do that all ISO for this video. Yeah. A little bit more. Yeah, it's alright. I'll just get a bit closer.
Oh wait, no, it's a ctenophore. It was messing with me. It was acting like a eel. No, wait, I don't know what this thing is. It's one of those two. It is a eel larva. It was just
Really-
glittering.
It's really see-through.
Yeah.
Kind of hard to.
Ooh.
Just look back. This is just an arbitrary waypoint. It doesn't really have to be going. I actually think they're getting pushed more to the east, but
Yeah, when I left, it's much southeast. I changed my hand to go with the.
yeah. Bridge, we've updated that waypoint just a little bit. I think we're getting pushed more to the east than to the south.
There you copy that.
Mm-hmm.
I'll look at it or not.
No, we saw a few of those before.
Okay.
Sometimes when the curve doesn't work.
Yeah, I know. I noticed that. This guy? No?
We could take a look at that one, yeah.
Yeah?
Yeah.
One more bump on the ISO. Yeah.
Nice.
Happy?
Yes, thank you.
Actually, more difficult to get those shots with this all the way out there. Because usually we'd have all that in. You can see what it is in relation to
Yeah.
the camera to get the depth perception
Mhm.
better. It's kind of hard to see it when it's
Makes sense.
there's all that stuff going on. You know what
Yeah.
I mean?
It's another one of those siphonophores.
Lift it.
Put him to following up on the lips a little bit. And then
Straight up to the umbilical.
Fish or worm.
Worm? But what's this other thing?
This thing.
Oh, comb jelly. Just different one. Or no, it's a siphonophore. Maybe they're tangled. Maybe there's two things. Oh. Yeah, it's like a siphonophore that's grabbed onto something. Oh. Oh, I think it caught an amphipod maybe?
Yeah, it's swimming right at me.
Really?
That's annoying. Oh well. Sorry, dude. Lost your meal. No.
Same one that we've seen.
Mhm.
A lot.
You're so jaded.
Oh, these guys again.
Something's flashing at us.
Yes.
There's like a fish that was like somewhere over there flashing,
Okay.
but it stopped. Oh, there it is.
Oh, yeah, I see it.
Mm-hmm. It's like a hatchet fish maybe or something.
Yeah.
Do the isochroma again, Leo.
Come on, man. Just look at you. I think that's a myctophid, so a lanternfish. Oh.
You just go right for the camera.
Yeah.
Get out of there.
Come on. Don't be in the shadow.
If you just stay still for a sec.
It's like it might get tired eventually. There's a couple of them now. Oh well.
It's not fast enough. Autofocus doesn't play games still.
It's transects, yes. Manual.
Does the autofocus struggle when it's just like one thing in front of
Yeah,
background?
it can in the mid-water especially.
Mm-hmm.
Just because of the nature of like you'll
Yeah.
be trying to follow something really little and then a big bit of
Mm.
particulate or something will fly across the screen and
Yeah.
then it'll lose its focus. And
Maybe that's not a McTophid.
see if I can get this.
It's also so shiny, it's like kind of hard to see.
It's a bigger fish here.
Oh yeah. That's funny. We've seen a lot of those. At least a few. Those bigger ones like that.
This guy's behaving now. His time off the screen.
Right, there's the other fish. Yeah, see, I think he's getting tired. Sometimes with the deep sea runs, you can just tire them out.
Yeah, he just keeps going so close and so
Mhm.
far, so fast.
That was pretty good though. I think it was
This guy's just
McTophid,
chilling
but
on our front porch.
oh my god, dude. Just hanging out. I have no idea what type of fish that is. I'm so bad with fish.
It's making our job quite easy.
I know. I should be able to tell. Definitely a shallow water one during the nighttime. Predator. She's gonna take a ride.
You can keep it within that waypoint as well. It's not really. I will just move it so he's got something to aim for, but we're not going to be stopping just yet. Interested in this? That's again another soul misses, is it?
Yeah.
No, click that again. I think it's not taking effect. There we go.
Mm-hmm. Oh.
If you're wanting in one of those jars, you go for it. That one if you want.
He's so close. It's funny, it likes to hang out on our porch.
Yeah. Like I'm coming up and down as well.
Yeah,
It's staying with us.
that's funny. It's like trying to get into a closed jar.
If it goes anywhere near an open one, it's coming.
We've seen this one, yeah.
This guy.
Mm-hmm. Another
Zoom
one
as well.
Yeah, another. Well, let's zoom actually.
Zoom.
Yeah. Might be a different thing. It's cool how it like glows. Wow. Yeah,
It's
that is a
tiny
different
little-
one.
Tiny little jelly just chilling. there.
Mm-hmm. Like it almost looks fluorescent. Wow. Really nice shots.
Bump up ISO a little bit, yeah. Give me a tiny little bit of wire out as well, Leo. Not a little bit of wire, just a bit. It's like pulling me a little bit. I know. Wire. Like that.
Oh, yeah, I thought you were saying whiter.
Yeah, me too.
Scottish accent, hard to understand something.
There's a lot of Scots on board.
Good
that
bunch.
just a coincidence?
Good bunch, the Scots. Very valuable to have on a ship.
Yeah. The amphipods.
Total bombing.
Seriously.
Amphipods again.
They just want to be in the shots. They're definitely attracted to the light. The longer we're in one place, the more show up.
Yeah. Done with
Nice.
this guy.
Yeah.
We're seeing fish.
Mm-hmm.
Fast moving fish.
Another jelly. Oh no.
Well, look at
Amphipod, yeah. Oh yeah, that's a hyperid amphipod. Nice. That's the type that colonize the salps and parasitize them. So that one's alone. They like scoop out the inside of jellies and use them as like little houses to travel around in.
Ooh.
They're the inspiration for the alien from like Alien.
I was about to say that's what it looks like to me.
Yeah, no, literally.
Geiger?
Yeah, he like got inspiration from these guys.
That's awesome.
You can see it.
Yeah.
Yeah.
That's all right.
We get a ton of them in the nets, but it's cool to see them alive and doing their thing in the water.
I'm trying to get out of my shot.
Mm-hmm.
Trying to evade me. I can managing to evade the autofocus anyway. Alright.
It's funny how they just go in circles.
I'm gonna lose him. It's a shame that all the focus doesn't like us too much.
It is like mostly transparent.
He's in speed mode.
Oh,
it's a
squid.
Okay, mate. We'll come and look at you.
I was like,
It's really
if we're
long.
already ready to look. Wow. Oh, that was a good little shot.
Squids always steal the show.
Yeah.
Okay, right. Got it on.
is this?
interested.
Oh, okay. So this is one of them that is actually inside of a salp. So it's parasitized it, but that's the same thing. And this one has laid its eggs inside. That's what the kind of orange thing is. You can see how their swimming behavior works a little bit better when they're inside of this. They don't just like
Yeah.
spin in circles as much.
He's got a bit of direction about him now, does he?
Mm-hmm. A little bit. Still kind of
bit.
aimless.
Well, but he's just not doing as much flips.
Yeah.
What's that guy called?
Phronema. It's the genus. Phronema. Yeah, it's a type of amphipod. Sorry?
I'm gonna take a stab at
It's a PHRO. I think it's PHRONEMA, but maybe I'm really bad at spelling.
We'll get there together.
Ooh, nice. Wow.
Okay, that was close.
So you know when you're zooming in,
Oh,
so
with an
you
eye,
just-
okay.
Just do it little bits at a time, so it's easy.
No, but this one I try to take the time when it's kind of still to zoom in and then
It's a really cool shot of the eggs. Oh. Nice.
It's tricky to keep in focus because they move about so fast.
Mm-hmm. There's another one just floating by. You can see how their arms are adapted to hang on to the inside.
I'm gonna go off the screen.
Nice.
Alex might like that. What's this little creature? Oh,
That's
not
a naked
bad.
one, yeah.
Yeah. All right.
Yeah, suddenly there's so many of them.
Yeah. All over the place now.
yeah, no.
So they only had to help me a little bit.
But also I was wrong because I suck at spelling.
I read a friend email. It's from email. Long boy.
Never jellyfish. Another jelly.
How those jelly jars holding up?
Good, but I don't think the glue inserts improving anything. I've got 4, but the glue insert like displaces the water a little bit. You know what I mean? So either make it bigger holes or just don't use it. It was just a theory.
What mesh size is it?
I don't know. We just 3D printed it.
Oh, okay.
It was 1.5 mil holes. But I mean, I don't know. We've managed easy enough without.
Yeah.
It was just an idea if something.
Now that they
was-
close faster, it seems
Yeah.
less necessary.
You're doing 600 psi on that?
I don't think so because the RAMs are 500 psi rated. So I don't know. Oh, were they? Number 1 was really slow to close. It was just this jelly, liquid jelly that hovered in there for ages and started going. So I'll try to get the same jelly in 2, and I got like above it and I pushed it away from the jar. You know what I mean? Just by displacing that water. So.
Looks like the latch hasn't stayed on that first one, so it'll probably drain when we get out of the water.
Hoping it's only the top because the 2 latches are separate. So hopefully the bottom one, because it was all set on deck.
Okay.
So I don't know what's happened. It's just
Vibration.
yeah, vibration in the current or something. I don't know. But yeah, I haven't even tried getting 6 because 6 was a little bit strange before.
Kind of like winter.
Yeah, we were waiting on that.
Yeah.
But yeah, we put it on while we've just been handing around at 500 meters just now. So
Good. There's fish.
fish.
Yeah, those have been hanging out on the porch. Just like chilling down there.
One of them almost self-sampled.
Yeah, got really close. I tried to swim into one of the closed jars. There they are. I don't know what kind of fish it is though, because I don't do fish usually.
What kinds of stuff do you guys already caught?
Today, soul missus, jelly, little tiny wee thing.
Sure.
Radiolarian.
The other one was quite small too. That's as far as my
Knowledge.
my knowledge
That's pretty
goes.
good.
Have we had
Yeah.
no worries? Little wee thing.
That's apparently a favorite
That's the way
area.
I'm gonna call the clip.
Yes,
It
perfect.
comes out of it. Little wee thing.
When they don't have a common name, that's
It
as good
works.
as you get.
This is just one of these guys
Mhm.
again. Yeah.
Yeah, so the faired area is in 3, and there's apparently a helmet jelly in 4, and then solstice is in 5.
Nice.
This was all before I was here.
Look at this chocolate. Ew.
Did that wrong.
Ew, get it away from me.
Where's your willpower?
Like willpower?
I,
What willpower?
My willpower would have to be too good.
Yeah, I was gonna say. Same.
I wouldn't. Yeah.
I've just been sneaking myself little bits of that milk and chocolate every day.
These fish are so funny.
He's a buddy.
Ready for it to go down the jar. Come on, dude. Just go in.
Try it. I dare you.
No, they just want to hang out on the porch.
Yeah.
Catch a free ride. Oh.
It's a good attack.
Oh, there it is still.
Yeah, here he is.
Oh.
Mm-hmm. We've seen a few of these.
Do that.
Still got more ink to go. Nice. Wow.
That's it. Yeah.
It's so
Oh,
beautiful.
let's go.
It's still got ink.
There's another one shooting off next to us,
Is
isn't
it? That's funny.
Come around himself if I can. I don't know if you'll let me.
Okay, no worries. I'm just, the camera's a bit zoomed in if you wanted me to.
Alright.
He's looking at us.
Oh my god, he's turning purple.
What go in the jar, buddy?
It's really cool how it holds its tentacles.
Oh, that's fine. Can I try and zoom in on his tentacles there?
Yeah, go for it.
So pretty.
It's turned.
It's It's like, no, you're not gonna see them.
You're not getting it.
Wow. It looks like velvet the way that the colors are.
Come on, buddy. Show me your arms. He's like, no.
Come back right the other way.
Just so turned away.
Turn around him a little bit the other way, so.
Thanks.
Well,
Oh, sweet.
nice.
Do a little scene while we do the
Wow.
Yeah.
Whoa.
Just keep an eye on my head and Leo took it all heading off. Fast.
Oh.
Yeah, the jar.
Almost went in the jar. Bye.
Bye.
That was great. There's still a fish down there.
That's still there.
No free rides, dude.
Yeah.
Sorry, that was super cool.
Yeah.
I needed that.
You need to.
Wow, we chased it down like 10 meters
Yeah.
more.
He was compliant for a while at least before
Yeah.
he decided.
Yeah. Yeah, it's usually quite rare, like, squids are
Yeah.
hard.
What's this? Do you know?
The pom-pom?
This little
Oh,
guy.
this thing. Oh, nice. Let's look at that. Oh, I think that's some squid ink,
Yeah.
right? Yeah. Maybe? Actually, I don't know. It's a weird piece of detritus. Could have been like the remains of an egg mass or something.
Focus is not what you're trying to do with this.
Or it could be part of a siphonophore. Huh.
I love it to be
We're just in like Phronema land.
Yeah.
So many of them.
Oh wow, there's a whole cluster of them.
Yeah. Like look at that. There's like 10 on the screen right now.
Is that normal?
Well, I think some of the other amphipods are attracted to the light, so I wonder if these ones kind of came over while we were hanging out there.
I see.
But maybe not. Maybe they're just in a swarm.
scoop up in the tube.
Mhm.
There we go.
Krill.
This fish is still hanging around us.
Oh my god, yeah. If you want, we can kind of keep creeping up towards 400. It looks like the density is sort of in between 500 and 400, so.
Sure.
But slowly.
Not slow.
Should we look at that fish? If it'll let us.
Sure.
Coming up.
Sounds good. What is that thing? Might just be more ink.
It's similar to the one that we saw.
What?
Similar to the one that we saw earlier.
Yeah.
Start stretching.
Yeah, so like probably ink, but it's kind of weird.
What?
No. We're just going. But we are moving with the waypoint moving.
Yeah, we can keep going. I don't really know what it is. I was thinking probably don't go past 400 yet, just because I think the current's going to get bad after that. And also between 500 and 400 is where the highest density of animals is at this point. Nice. A couple of siphonophores.
Really, if you look at
What? Nice.
How
So
do
that's-
you read this?
Yeah, so these are different wavelengths. On the left is going to be the biggest wavelength, so it'll detect the largest things, but otherwise it's the same, so going down in depth. And the darker colors is where there's more animals. So in the largest size fraction, the animals are mostly around like maybe 350, whereas this next size fraction, they're closer to like 450 to 500,
So the dark area is where is the...
where there's more.
I thought
Yeah.
it was the opposite.
No. Or no, sorry, the brighter is where there's more.
Oh, okay.
Yeah.
Okay.
And then this is like an artifact because we're not moving, so it's not going to be, like, it's not the kind of data we would be able to like quantify. It's more just useful for where to target.
Okay.
That's like, I guess I don't know if that's the RV or what, but there's something there.
that's the ADCP there.
Oh, okay.
38 kilohertz ADCP and 38 kilohertz VK81.
There you go.
Zebra.
Yep. Do you know what size fractions it's showing us? Or I just know it's like larger to smaller, but I don't know like what that would actually correspond to.
Yeah, but the calibrations, like you said.
But you can see the migration. So I mean,
The
we
yellow
stopped it,
thing.
so it didn't go straight up and then down, but you can see the down, yeah. Things went back down.
Yeah, I think early this morning was more concentrated, lower than...
Yeah. Yeah, when we went in at the beginning of the dive, stuff was really at the surface. Oh, it's a really big larvation house.
Yeah.
You want to go first? Still hanging out below 400.
Okay. On now.
Ooh, let's look at that thing over there.
That thing.
Yeah. I've been trying to figure out what they are. Because I think it's a ctenophore. Yeah. Nice, definitely ctenophore.
Sorry, what is it?
That's a comb jelly. It's a ctenophore. It's just like a really weird one where they're long and flat. Seems like there's a lot of them at this station. Usually that line is kind of in the middle, so I don't know if this one, like, part of it got broken off. Wow.
Let's do it then.
No. Okay, yeah, I think that, yeah, the genus is probably cestum. Someone in the chat mentioned. That was great, though. Oh, and there's a fish right there, too. Let's see if we can sneak up on it. We definitely got some of this kind in the nets, but I don't know what kind it is. It's funny how they just go down to try to escape. Bye.
Why not do that?
I'll go, Red.
Yeah, another scythe.
Oh, you'll be able to bring this up. That's right.
Fish.
Where? Oh. Just hanging out.
Oh, fishing.
They've been hanging out there. It's so funny.
He likes it there.
Yeah.
Look at this little house. It's gonna be real disappointing when they come in.
I wonder if it's feeding on the amphipods that are attracted to the ROV. Well, they don't usually feed at this depth, but it might be opportunistically feeding. Let's look at this siphonophore.
There's a lot more stuff here.
Yeah. This seems like the big daytime layer.
Zoom in.
Ooh, yeah, that's crazy how it's just like shedding a bunch of goo and bits.
Yeah.
Wow.
There's a lot of ingredients, yeah.
Yes.
I wonder if it's shedding its tentacles as like a defense thing to have predators go after that. But I don't really know. Nice zoom.
Close to the
Yep.
relative decision.
Thank you.
That's a cute little jelly.
Hmm?
Right in the middle. Mm-hmm.
Mm-hmm.
Too fast. You can see this one's a little older. It's got that like teardrop shape. There's definitely like little copepods on it, eating it as it sinks.
Are there other things that eat larvaceous
Yeah, definitely a lot of like protists. And probably amphipods would as well. They just have to be able to kind of sync with it, and then because a lot of them aren't going to be able to withstand the whole pressure gradient all the way down to the seafloor, so they might just be on it for a bit and then leave. And that's pretty.
Mm, that one looks fancy.
These are not the camera. Come back to the left right now.
Oops. It's a lot harder to get these like super transparent things exposed.
Oh, our jelly's still alive in the first jar.
Yeah. Here, you have.
Mm-hmm.
It's just that you haven't done that in a while. Fooled me. Stay on my toes all the time. Aren't you?
What's that below us? Oh, another scythe. Mm-hmm. The fish.
Ooh, big fish. What did you say?
Urinary appendages.
Little.
Oh, I think that guy's got a parasite. Oh, don't swim into the siphonophore. So transparent. Wow. I think that's someone else up there.
Nothing beats lava.
Go.
I was in this chocolate.
No, thank you.
Think, can I have some?
Of course. So.
Oh, two squid.
Oh my goodness.
Come on, guys. 3. Oh my god.
Oh.
So many of them.
Yeah.
Must be a school. Those the squid have not stayed calm.
That still doesn't.
Now we can't see anything.
Push through.
Is this what it's like to be a predator?
I think so.
Oh.
Gross.
Now you know what your dog sees when it gets stumped.
At least we don't have to taste this ink like someone else would. It probably does not taste good. Although I guess people do eat squid ink, so maybe it's okay.
No, it doesn't really have. It's kind of red, right?
Yeah.
Not much of a flavor.
So it's 7:35.
Uh-huh.
I don't know how much time you guys need, but we could like start very slowly heading up, or we can wait. It's
could start coming up slow, but we're gonna need, when we hit about 200 meters, we're gonna need all 4 of us to
Mm.
out on the
Okay,
winch
so we
and
can
stuff,
wait.
So, I mean, we can.
Oh, 15 minutes.
Yeah.
We'll just mosey up.
Cool.
Just meander.
Perfect.
Right, Jarvi.
Yeah, go ahead.
Morning, mate. Would you be able to give us more of a northerly heading in preparation for us coming back to deck? I believe we're going to start ascending after the morning meeting's done.
Yeah, that's a good point.
Yeah, mate, thank you. Are you expecting to see much in this last few hundred meters?
Not really.
Yeah.
Seems like there's a lot of stuff like within 100 meters, but you know, we can't really hang out there, so. Oh, what is that? Somebody just floated off to the right. I think it went to the right again. Maybe up. Maybe not. Looks like a fish.
Mm-hmm.
a worm.
It's a wormfish.
I think it was a worm. Yeah, this is like suddenly the Ketignath area. Yeah. Oh yeah, what's that? Just in the middle of that screen. Oh, it's just a larvation, never mind. Something sizable. Probably also a larvation. It's weird how it's like stretched out so much. But I think that's what it is. Larvation. Mm-hmm.
Hello, hello.
Loud and clear on me.
Okay, I'm out here already.
Copy.
Bridge, ROV.
Thoroughly bridge.
Yeah, can you inform the deck guys that we're going to be coming on deck pretty soon?
Sure, no problem. Now is here.
I know we're speaking to you on comms, Schmidt. You already announced yourself.
The other.
Why are you speaking?
Are you speaking about yourself in third person, mate?
Yeah. My alter ego.
Has the same name as you.
cruises typical to have like a 13-hour dive? Is that something you do frequently?
Yeah, we can do longer.
I
Mm-hmm.
was gonna say, we've had 50-plus-hour dives before.
Wow. Probably not collecting samples.
No, that's why we come up, because we're full.
Yeah, okay, yeah.
In the Galapagos, it's really comforting.
Yeah, otherwise we have 21 day dives.
What?
21 day dives.
For real? So it can just sort of stay down there indefinitely. It's
Yeah,
just about.
not this vehicle.
Oh, okay. Other ones, yeah.
There's certain things, if everything is going right, then yeah, it can stay in.
Mm-hmm.
Indefinitely.
Gotcha, there you go. Oh, coming up.
We are now.
Huh?
We were hanging out at 300. Just waiting for you to come back.
Okay.
Before we start coming back, we're gonna put me in back.
I'll start hauling in.
Yep, coming up.
Happy with everything. The only other thing that we were gonna do before we reached was just make sure the jars that have anything in it are just super closed. Perfect. Thank you.
Coming up. Put me into.
Once we are on deck and everything, we'll open the tops for them. Because I think last time somebody pushed the hole in and emptied all the water out of the one that held water. That's a big old creature you got one, huh? That's cool.
Big old jelly.
Big old jelly.
Hey, we're coming up, Miles.
Yep. Can hold the control if you want.
Okay.
You got it.
Okay.
You got it? Okay.
Loud and clear, help me.
Roger. Permit.
Good, how are you?
Okay, can I come up?
Coming up.
I'm gonna go ahead and put you in launch and recovery.
Sure.
Andy. I'm going to do that as soon as this process is finished. However, I fumbled with it yesterday, so I'm gonna have to select the display. Then do I have to hit quad? Quad settings. And I want to change number 4. Yep. And I want to do ROV. And I want to do SD.
I think it's 2. SD2. That's it.
Thank you for your support.
Anytime.
Yeah, I was like, I did it a couple. I'm like, why does this one just keep changing? I'm on the wrong quad.
Yeah, I think I need to update that preset.
Do you want to hear
And hello, everybody. Good morning from the control room of Falcor 2. We are just finishing up this dive. You probably heard us handing controls over to the deck. And as always, I'd like to thank everybody for joining us today, for all your wonderful comments and questions, and for supporting us. With that said, if you want to follow along on this expedition, feel free to follow us on social media at Schmidt Ocean. And hopefully we will be back sooner rather than later for another dive. Hope y'all have a great rest of your day. Enjoy the rest of the ascent, and we'll see you all next time.